once the action potential reaches the myofiber, the muscle fiber, and the action potential is generated in the myofiber and it propagates down that transverse tubule and it releases calcium from the sarcoplasmic reticulum, the next thing that's going to happen is the formation of something known as a cross bridge. And the cross bridge is where myosin and actin are going to interact with one another. And so we have to remember that actin was this globular protein that became filamentous, right? So multiple G-actins came together and made this filamentous actin. Okay. And if we recall that there was a filamentous protein that wrapped around that actin filament, like so. Now, if we sort of blow this up, what we're going to see is that if we look at, there's a globular actin, there's a globular actin there. What we'll see is that there is a specialized site on this actin filament, and I'm going to sort of highlight that. Let's do it in purple, actually. I'm going to highlight that in purple, like so. Okay, and that site is the myosin binding site. So myosin has a high affinity for actin, loves actin, right? Wants to bind to actin, but it can only do so in a very specific spot, that myosin binding site. But under sort of a resting state, that tropomyosin sort of wraps around like so, and it blocks that myosin binding site. Now, there is another protein that's attached, and it's this 3 subunit protein here, this trimeric protein, right? And that trimeric protein, remember, is troponin. Okay? So the troponin, right, is sort of attached to that tropomyosin. So there are these 3 subunits for troponin. So we look at troponin. There are 3 subunits, and they are TNT, TNI, and TNC. Now, TNT binds to tropomyosin. So that's the one that is sort of attached to that tropomyosin, right? So this would be the T subunit here. Then there's TNI, and this is one that binds to actin. It's known as the inhibitory subunit, right? So this would be TNI here. And then there is TNC, and this one binds to our good friend calcium, right? So this would be TNC there, right? So troponin has those 3 subunits, okay? And then up here, we'll just sort of draw our big myosin globular head, like so, right? So what's going to happen is that calcium is going to come in and it's going to bind to that calcium binding subunit of troponin. And what that's going to do is it's going to cause a conformational change in that troponin. Now, because troponin is attached to tropomyosin, it'll also cause a shift or a pull in the sort of tropomyosin. So when we originally had this conformation up here, right, what it'll do is, if I sort of just draw this generically, right, is we will take and we will shift it so that it sort of spins along the axis and looks more like this now, right? So this portion here, right, spins around and sort of flips around sides. So what happens if we look at our blown up model here is that you have your actin, right, is that troponin will undergo a conformational change, and what that will do is it will wrap the actin or move that tropomyosin. So we'll have our 3 subunits here. Right? So this causes a shift so that now that myosin, right, can come down and it can attach to that myosin binding site on the actin filament. So what's going to happen then is you will actually get this attachment of myosin to the actin filament. Right? So now these two are connected, and that's that formation, right? That connection point. That's what we call a cross bridge. So the cross bridge is the connection between the actin molecule and the myosin molecule. So it is, right, the movement of the tropomyosin that exposes the myosin binding site that allows it to bind to actin, but that is controlled by calcium, right? So when calcium is released from the sarcoplasmic reticulum, right, it's going to bind to the calcium binding subunit, causing a conformational change in troponin, which pulls on the tropomyosin, exposing the myosin binding site on the actin filament. So let's go ahead and let's just write those steps down, right? So the last time we talked about steps, we ended with calcium being released from the sarcoplasmic reticulum. So now we have calcium inside of the sarcoplasm is going to bind to the calcium binding subunit of troponin. And that is going to lead to a conformational change in troponin. And it's the conformational change in troponin that will lead to the conformational change in tropomyosin. Okay, or the spinning of that tropomyosin around the actin filament. So that's going to expose the myosin binding site on actin. And because myosin has a high affinity for actin, myosin binds to actin. And it's at this point, right, we'll put in parentheses, that is the formation of the cross bridge. And that's how we get the formation of the cross bridge. So in the next video, we'll talk about the actual movement of the muscle itself. Woohoo!